Hello, everyone. Welcome. My name is Kaylee McCauley. I'm a registered dietitian and health promotions and research manager with Celiac Canada. I'll be your host for tonight. We're so glad that all of you could join us. We have over 500 people registered, so it just shows how much interest there is in learning about the gut microbiome and celiac disease. Just before we begin, a few housekeeping notes. Tonight's session will be recorded, and the replay will be available for those who registered. Please do not distribute the slides afterwards. The Q&A box is on the bottom of the Zoom, and you can put your questions there. We'll take as many as we can at the end. If you experience any technical issues, feel free to send a message in the chat, and I can try my best to help from there. A big thank you to our premium sponsor, Odos, whose generous support makes this webinar series possible. Odos is dedicated to supporting the gluten-free community with safe, nutritious, and delicious products from breads to buns to flatbreads and more. So now I'm thrilled to introduce our speaker, Dr. Elena Verdu. Dr. Verdu is a professor of medicine, Tier 1 Canada Research Chair, and director of the Farncombe Family Digestive Health Institute at McMaster University. Her research focuses on how microbes and diet interact to influence conditions like celiac disease and inflammatory bowel disease, IBD. She is a world-renowned researcher, a past president of the Society for the Study of Celiac Disease, and has received numerous awards for her groundbreaking work. We're so grateful to have her here tonight to share her expertise. Dr. Verdu, welcome. Thank you so much for joining us.
Thank you very much for the introduction, and I'm very happy to be here today. Hello, Canada. So give me a few minutes to share my screen. Okay, can we all see the slides? Then we will get started. So the microbiome and why should we care? And I put it between brackets in celiac disease because I think in general we should care about our gut microbiome. But let me start with some definitions. You will see these words over and over in the lay press, and we really need to understand what they mean, what we mean by them. So microbiota, the microbiota, biota means life in a way. So it relates to who is there, who is in our gut. And these are a collection of microbes from bacteria to viruses, fungus, there can be even some parasites. And when we speak of the microbiota in general, at least during this talk, I will mainly be talking about the bacterial component. This is the part that we know most about, and this is the compartment that we actually test when we do our microbiota sequencing. So microbiome, it's not the same as microbiota, although some people use it at the, as interchangeable terms. Microbiome is more general. It is a higher level. It means the collective number of genes of that microbiota. So it is not who is there, but it's what can they do? Because two bacteria can do the same thing, and they can be very different. And then we are going to talk a lot about microbial metabolism. So this is more functional term. It is the activity by which components of the microbiota can break down molecules inside of our gut. And this can be food stuff like gluten, amino acid, fibers. We are going to talk a little bit about that. But also molecules our body produces, medications, toxins that we are exposed to. The microbiota is able to metabolize many, many compounds, better than our liver. And so what does that microbial metabolism do, which is done by many microbial enzymes? They create more molecules that we call metabolites. And many of these metabolites are going to be very active. They can actually activate some receptors in our gut lining, and modify functions and immunity. And so some of the metabolites we are going to mention are indoles or short-chain fatty acids. And we are going to speak sometimes also about gluten tolerance, gluten intolerance, what that does mean. This is a very actually in immunology, a very specific definition. And these are immune mechanisms that prevent that your immune system overreacts to molecules that should not create actually an inflammatory reaction. And so gluten tolerance are those mechanisms that protect us to overreact. And in celiac disease, we will talk about loss of oral tolerance. So definition of dysbiosis, because you will hear about this quite a bit. Oh, dysbiosis, I have dysbiosis, or such, or this food creates dysbiosis. So it is very difficult to define this term, and I prefer not to mention this word, but rather to talk about the changes that I find when I, for example, investigate the microbiota in a specific disease. But I like to depict this concept with a jar of jelly beans. You could imagine that jelly bean that we see here in the slide is the normal microbiome, with every jelly bean being a different color and a different bacterium or group of bacteria that behave similarly. Now, during your life, there can be exposure to infections, to drugs, and especially if we overuse antibiotics, and that can perturb, can decrease our microbiome. Sometimes you will hear about microbiome diversity is decreased. Now that's normal, and usually in a healthy person, the microbiome will recover. Now, if you have certain genes, you're exposed to other diseases, if you have medications or simply other lifestyles that could impact in a chronic manner that microbiome, then we don't recover. So that state of chronicity, of altered microbiome that associates with a disease component, then we could call that dysbiosis. And so what happens is that we have bacteria that live in a mutualistic condition with our body. And so some of those bacteria in certain contexts of inflammation could behave in a way that promotes further inflammation. But the gut microbiome in general is essential for health, and not only in the way the gut moves or propels our contents in the gut. It also protects the mucosal barrier because it produces molecules that will protect this barrier. It also instructs the immune system. And most recently, work from the McMaster University has shown that bacteria can even affect organs beyond the gut. And for example, there is something called the gut-brain axis or communication, in which the gut microbiome could influence through neural, through immune pathways, the way we perceive things. And then we are going to talk about the important role of the microbiome in metabolism. And I mentioned that they can modify or metabolize certain medications, certain toxins, but they also produce some vitamins that are essential for our health. It can also regulate energy and fat storage, and of course, it will metabolize food. And it will metabolize, when we think about metabolism of food by the microbiota, we usually think about metabolism of fiber, because fibers are carbohydrates that our enzymes cannot digest very well, but bacteria can do this, can ferment these carbohydrates and will produce metabolites that are important for our gut health. And gluten, which is not a carbohydrate, it's a protein, will also be metabolized by the microbiome, as I will show you in the next slides. So you can understand how, if this ecosystem of microbes in our gut becomes dysfunctional, many of these functions are also going to be impaired, and this could predispose to inflammation or disease. And so this is what happens. As I mentioned, we exist in a very delicate mutualistic relationship with the microbiome, where we both benefit. We benefit because we have a better immune system. We get certain metabolite functions that we wouldn't have without that microbiome. And the microbiome benefits because they are commensals. What that commensal mean? It means that you sit at the table and you share a meal. And so our microbiome shares the same food that we are ingesting. And so there are many factors that could alter, as I said in the previous slide, this mutualistic relationship, like infections, our genes, the food we eat, or overuse of antibiotics. But the reality is that that mutualism can change to a disease-promoting microbiome with the presence of opportunistic pathogens that could promote chronic intestinal inflammation. And so one of the main determinants of our microbiota is really diet, but our long-term diet. And so a lot has been said about the recent change in eating behaviors. For example, the Western diet, which is low in fiber. If it's low in fiber, then our bacteria get starved in the colon because they don't receive these complex carbohydrates, and then those bacteria that are fiber degrading are not going to be able to proliferate, and then we will lose this beneficial bacteria. This Western diet is also high in refined starches, low in fermented foods, high in additives and artificial sweeteners. And then we can think of the gluten-free diet, not the one that is a natural diet, but the gluten-free diet that is significantly based on ultra-processed foods that can also be low in fiber because of this overuse of the processed counterparts that are gluten-free. So long-term diets can affect significantly the composition of the microbiome and also the function of the microbiome. So this takes me to celiac disease, which at the beginning of my career, I postulated that it was a model to study diet microbe host interactions, because we know exactly what the trigger is, or at least the main dietary driver, which is this complex group of proteolytic resistant proteins called gluten. We know what the main genetic determinants is, which is to have HLA-DQ2 or DQ8. And as you well know, the prevalence of celiac disease is about 1%, but we have also observed that there has been an increase in prevalence very rapidly, which tells us that there is something in the environment that is promoting these diseases, not only celiac disease, but other autoimmune conditions and some allergic conditions. And so we have always suspected that there could be an environmental component that promotes this increase in prevalence. And indeed, there have been for many years this suspicion, because there is a clinical observation of some onset of celiac disease after gastroenteritis episodes. Also, physicians have noted for several years that there are antibodies in the blood of individuals with celiac disease that are increased. These are bacteria, antibodies against bacteria. And when these patients go on a gluten-free diet, sometimes these antibodies go down. And more recently, with the sequencing era, we have seen associations with infections or microbiome changes in at-risk populations that then, during the follow-up period, develop more frequently celiac disease. So all this is pointing at perhaps a role of microbiomes as the key factor or a key factor in celiac disease. So it's not what causes celiac disease, it's not the main driver, but it is an adjuvant cofactor. And so we have done some work during already the past 20 years on the role of small intestinal microbes as cofactors in celiac disease. And we have focused from the very beginning on gluten metabolism. To tell the truth, I must say that when we started in my lab with this hypothesis, we got a lot of backlash, and it was very difficult for us to sometimes publish or obtain grants to pursue this hypothesis. And so we took it very seriously that we really had to find very hardcore mechanisms to, if you wish, convince or provide proof that bacteria in the small intestine could have a role in celiac disease. And so we set out to study gluten metabolism by opportunistic pathogens from celiac patients, but also by commensals. And I'm going to talk a little bit about that. We did some studies on the regulation of gut barrier by microbial metabolites that were produced by the microbiome. We didn't do work on gluten tolerance, but this was work that was done by the group of Bana Jabri, mainly related to viruses and how pro-inflammatory viral infections could promote that breakdown of tolerance that I mentioned in the very first slides to gluten. And then finally, and after we published our papers in 2016, the same opportunistic pathogen that we had discovered played a role in incomplete gluten metabolism. A different group found that our immune system can recognize gluten and some molecules in this opportunistic pathogen. So if you wish, our immune cells may get confused when they see one or the other because those regions in the bacterium are similar to regions in gluten. So this is what we call molecular mimicry, because the opportunistic pathogen is mimicking gluten in a way. So going back to gluten metabolism, which was our first hypothesis of how bacteria could influence celiac disease, this is work that was done by Alberto Caminero, who was a postdoctoral fellow in my lab, and that today is an associate professor at McMaster University. And so what Alberto did was to start to characterize and isolate microbes from the oral and also duodenal cavity, as well as the stool, the feces, from patients with celiac disease and healthy controls. Now, I have to say that Alberto was one of the first to focus on the upper gastrointestinal tract. Now, I know you're all saying, but that's, of course, this is celiac disease. You should study the bacteria that is in the upper GI tract. But that was not the case. Everybody was using feces to identify microbes or to detect changes in the microbiome, and they were forgetting about the niche of bacteria that are very, very different in the duodenum than in the lower GI tract. And so what this slide shows there is actually a plate where we grow bacteria. So we get some bacteria and we plate it in that agar, and that agar, which is the lawn of the plate, contains gluten as the main protein source. And so bacteria start to grow, and you see three lines double, and what you can see is that the bacteria are in the middle, and then you see this halo, this gray halo, where bacteria are eating the gluten in the plate. And so this is the way we know, and we can isolate different types of bacteria that actually consume gluten. And then we can do our experiments with these bacteria. And so one of the first experiments that Alberto did was to digest gluten peptides with human enzymes. You very well know that our enzymes cannot fully break down gluten. And so then he added bacteria. He added celiac bacteria, that you have it there in green. He added healthy people bacteria that for didactic purposes are in blue. And then he added some gluten peptides. And he used a machine, it's called LCMS, that can tell us what peptides are being broken down, are being produced by that metabolism. And what he found was, and the Pac-Man figures are the enzymes of the different bacteria. So our human enzymes in red would not break down completely gluten peptides. We had peptides like the 33-mer. In green, you have the celiac bacteria. This was Pseudomonas aeruginosa, an opportunistic pathogen that we found in the duodenum of celiac patients. And the little green Pac-Mans are eating the 33-mer and breaking a little bit those peptides, but you still have big, large peptides that are not completely broken down. Now, the moment he incorporated in this mix the blue bacteria, which are the lactobacilli, and the blue Pac-Mans are the enzymes produced by the lactobacilli, then the LCMS had a flat line in many instances, and so we could say that this healthy bacteria could complete the digestion, and now we had detoxification of gluten. So this was the first really seminal study from our group that put small intestinal bacteria and the community, the functional interaction between the different players that are in the small intestine of celiac patients as a possible cofactor with a very clear mechanism that could influence celiac disease. Of course, we didn't know whether these peptides were stimulating the immune cells in celiacs or not. So we did further experiments. What we did was the same mixtures with human enzymes, gluten, celiac bacteria, or bacteria from healthy people. And then we got our metabolites, what we thought were going to be either immunogenic peptides by the celiac bacteria or detoxified peptides by the healthy individual bacteria, so the lactobacilli. And then what we did is we recruited celiac patients that were TQ2 positive. They were on a gluten-free diet, but they were given a gluten challenge. And then we can, in that way, stimulate the immune cells that are in the gut and move them to the circulation. And then we can take these immune cells and challenge them with the metabolites produced by the different incubations of gluten with the celiac or the healthy bacteria. And this experiment confirmed that if we only have celiac bacteria, then we have a proliferation of these cells. These are really gluten-reactive, gluten-specific T cells that produce interferon gamma. If instead we do this metabolism, adding the healthy bacteria, the T cells of these patients do not react as much. And so this really told us that the peptides that were produced by the celiac bacteria induced an immunogenic response in celiacs. And that if we had bacterial digestion with the lactobacilli, we had a lower immune response. So the microbes that we have in the small intestine impact the way we digest gluten. And so the next question was, well, what about bacterial metabolism of other compounds? There are so many metabolites that can be produced by this activity. And one that was at that time emerging was this amino acid tryptophan. Now, tryptophan is very interesting because we have to consume it with our food. And our human enzymes can metabolize tryptophan, and indeed it has many important functions, specifically in mood and neural regulation. But we also have microbial enzymes that metabolize this essential amino acid, and that's a completely different pathway. The metabolites that are produced by microbial metabolism of tryptophan, some of them are called indoles, and these are anti-inflammatory molecules that had been shown that are decreased in inflammatory bowel disease, so Crohn's disease and ulcerative colitis. So we asked, well, what is the role of this microbial metabolism in celiac disease? And so we set out to investigate this. And here what we are measuring are the total indoles in the proximal gut. And we see that patients with active celiac disease have a lower content, so the same that had been observed in IBD. And if we would use mice and give them gluten, these are mice that are genetically predisposed because they have a transgene that is the DQ8 molecule. And so if we give gluten to these mice, they develop small intestinal inflammation. If we give lactobacillus, not any lactobacillus, but lactobacillus that we know can consume tryptophan and produce indoles, then this inflammation is dampened in the mouse model. And this was a very interesting information. And we said, okay, so if we give now tryptophan to these mice, do they start to produce indole? Do they start to, the microbiome, does it start to change, to metabolize the tryptophan, and does it produce indoles? And yes, the answer was yes. Not only that, but when we measure the microbiota in these mice, you see up there in what I have highlighted in red, that the mice that received the high tryptophan diet change their lactobacillus abundance. The lactobacillus blooms because now it has something to eat and to produce indoles. And so the next question was, can we do this in humans? Can we actually activate the microbial metabolism of tryptophan in the small intestine? So we designed a study where we gave tryptophan to patients, but we wanted to release the tryptophan in the small intestine. So this is the stomach and the small intestine. We wanted release in the small intestine, so locally, to see whether microbes could break down tryptophan and produce indoles. And so we did this, what is called a crossover study in healthy subjects, where we administered tryptophan or placebo, and then we crossed over. The ones that got tryptophan first, then got placebo, and vice versa. And the answer is yes. This is the first demonstration in humans that if we give tryptophan, we can increase the production of indoles. And this happened in blood and in urine, but not in the feces, which suggested that really this metabolism was occurring up in the small intestine and not necessarily in the lower GI tract. And what was more interesting is that we had a way of measuring the activation of the human receptor for indoles, and we saw an increase in that activation, probably because these indoles were reacting with a receptor and activating this pathway that has been shown to be protective for gut barrier. And again, we saw no activation in the lower GI tract, so the way we were delivering the tryptophan with this release in the small intestine was really targeting the small intestine. And so we have actually concluded very recently a study in celiacs, and we are frantically trying to analyze the data to see whether we can also stimulate the pathway in patients with celiac disease, which we showed before that is impaired. And so maybe next year I can give another talk about whether we achieved that outcome in our celiac patients. I want to move on now to breakdown of fiber in celiac disease. Now, one of the most important and probably most studied metabolic effects of the microbiota is the metabolism of fiber. Fiber is the global name that we give to these complex carbohydrates that we can actually take as supplements, or we can eat foods that are rich on fiber, and we cannot break down fiber. But when it gets to the lower GI tract, it gets digested and metabolized by the microbiome, and it produces many important and beneficial metabolites like short-chain fatty acids. Now, when the student, the PhD student that worked on this project came to my lab, I said, but fiber is digested mainly in the lower GI tract. Why would we study fiber in celiac disease? But he insisted, and we started looking into this to see whether really fiber, some type of fiber metabolism could occur in the small intestine. And so we started a study that included healthy controls, patients with newly diagnosed celiac disease, and patients with treated celiac disease. And you will see the results will be color-coded. So healthy control results are in blue, the active celiacs are in red, and in green you will have the results from treated celiacs, which means 2 years or more on a gluten-free diet. We measured fiber-degrading microbes in the small intestine. We also did it in the stool, but the novel part here was to look in the small intestine. And of course, we investigated dietary patterns. We wanted to estimate whether these individuals were eating fiber and what amount. And so we did this with a typical food frequency questionnaire, but we also used an objective test, which is called food sequencing. So in the same way that you can sequence your microbiome, this is a way of sequencing the DNA of the food that an individual eats. And so this is very objective and not as biased as a food frequency questionnaire. Honestly, I don't remember what I ate for breakfast today. So it's very unreliable sometimes to only base on the recall of the foods that you have ingested. So let me start by and this is all unpublished, so please don't post this anywhere. The manuscript is currently in review in Nature Communications. And so the fiber-degrading microbes that we discovered, and this is a very big name, you don't need to remember that, but simply know that this is a family of bacteria, Prevotella or Prevotellacea, that are very well-known fiber-degrading. And so we found these bacteria that were very prevalent in the small intestine. And as you can see, both active celiacs in red and also celiacs that were on a gluten-free diet have lower amount of these family of fiber-degrading bacteria in the small intestine. We were a little bit surprised because we were hypothesizing that because the gluten-free diet has been associated with being low in fiber, that group, the green group, the treated celiacs, were the ones that would have a depletion of these bacteria. But we saw that the active celiacs already had that depletion, even when we detected that they didn't have significant differences, at least through the fiber estimation, the food frequency questionnaires, on the daily intake of fiber. There are no differences, statistical differences between the three groups, although most of the individuals, these are small cohorts, were below the Health Canada recommendations for dietary fiber, which is not surprising in the treated celiacs. But we had individuals with very low fiber consumption among the healthy controls and also in the active celiacs. So the fiber intake was not explaining this depletion of fiber-degrading bacteria in the celiac population, either active or treated. And when we did the fecal plant DNA analysis, we found no differences in plant richness. Not at all. There were no differences between the three groups. The only difference was that the treated celiacs on the gluten-free diet had a shift from their fiber source. Instead of being wheat, rye, barley, it was mainly rice and potatoes. But apart from that, there were no differences that would explain this depletion of bacteria. So we think it is related already to the inflammatory status. Now, what is interesting is that if we measure the genes that are actually related to fiber degradation in that family of fiber degrading bacteria, the Prevotella, we did see exactly the same, a depletion of this collective gene capacity for degrading fibers in the celiac patients, whether active or on gluten-free diet. So this is the first report to our knowledge that is showing that small intestinal celiac bacteria are less efficient at degrading fiber in the small intestine. Of course, we also found the same in feces, but what is important is that then there is a lower production of these protective metabolites, those short-chain fatty acids that I mentioned at the beginning of the talk, specifically in the small intestine. And so the mucosa is depleted from these barrier protecting metabolites. So this is just to open the discussion. I know that I gave you a lot of information and you may have many, many questions, but probably the takeaways that I want to leave you with is that we need to study the small intestinal microbes. And indeed, I think that our studies have really contributed to put intestinal microbes from the upper GI tract on the map. They are gaining attention in gastroenterology, and not only in celiac disease, which is the obvious small intestinal disease, but also in conditions like irritable bowel syndrome and Crohn's disease that can also affect the small intestine. So in celiac disease, I think we have made tremendous progress in terms of identifying specific microbial metabolic deficiencies in the microbiota of celiac patients, and they relate to gluten metabolism, which was our first studies in this area, tryptophan metabolism, and fiber metabolism. And what is interesting is that very recently, I was very happy to see a study from the TEDdy cohort that actually shows, and this is just association, there's no mechanism to explain this, that more fiber in infancy in these at-risk children is associated at the follow-up period with lower celiac onset. So really a clinical explanation or consequence of the mechanism that we are starting to investigate in terms of fiber metabolism and celiac disease. So why is this important to know all these mechanisms? Because they give us the opportunity to really optimize microbial function in the small intestine. And we can do this not with a drug, but through dietary or probiotic supplements, and maybe better manage and control the disease. And so while this is quite obvious that we can do this, the obvious question is also when. When should we do these interventions? And this was very nicely actually discussed in the last Tampere meeting in the Symposium of Celiac Disease, where, well, our microbiome, we get it through maternal transmission. We get it from mom and dad. It can be influenced by the genes we have, but is significantly influenced by the dietary patterns we have and our lifestyle. And so that microbiome is very important for maintaining a good gut and overall health function. Now, as I mentioned, there can be infections, diseases, drugs that we need to take that will affect and deviate that microbiome, what some people call dysbiosis, and which is not the same in two different patients. And that, in the presence of certain predisposition or context, can promote chronic inflammation. So if I show you this slide, you probably say, well, we should prevent these changes by intervening at the early stage, before these changes happen. In other words, to make our microbiome more resilient. But unfortunately, as adult gastroenterologists, we usually see patients after they have developed these changes. And so we have to also develop treatment opportunities when these changes have occurred. And the last question that I think many people forget is how to intervene. And again, during the Tampere Symposium, there was a very nice presentation from the TEDdy cohort, and they were showing now we could take these individuals, these children, and follow them up and intervene with a probiotic. And when I asked them, what probiotic are you giving? How did you choose it? Oh, it was the one that we could get off the shelf in our country. Honestly, I think that this is a mistake, and we are going to be disappointed if we do these blinded interventions. We need to investigate mechanisms. We need to understand microbial pathways in the disease we want to treat. And then we need to select a strategy that targets that pathogenic pathway. So it is a long process, but it can offer a better outcome. Then we can do an informed intervention or clinical trial. And I gave you here 3 possible metabolic pathways that we could either individually or in combination exploit to develop a clinical trial that we have more chances to succeed. And so with this, I would like to thank everyone. I know that it's late in some parts of Canada. So thank you for staying until the end, and I will be very happy to answer questions. Oh, I see that there are many questions already. So would you like to read them, or how would you like to do this?
Yeah, I can read them, or if you'd prefer, you can do it as well.
So there is a question, is there research about how folks eating a strict gluten-free diet still have an overgrowth of bacteria? Okay. Yeah, so SIBO, or small intestinal bacterial overgrowth. This is a condition that it's sometimes overestimated because we don't have a good way of measuring SIBO. We use tests that are quite imprecise, and sometimes we overestimate it. And the treatment for SIBO is antibiotics. And sometimes the patient does not get better, and they get a second turn of antibiotics. And it helps for a little while, and then again another round of antibiotics. And honestly, this is going to make things worse in the end, because as I said, overuse of antibiotics can lead to a significant chronic change in your microbiome. So we have to be very careful of whether SIBO is a real entity or an overly estimated diagnosis. So I would first want to know whether SIBO has been properly diagnosed. I hope I have answered that question, but there is no evidence that a gluten-free diet would induce small intestinal bacterial overgrowth. What is related to maybe small intestinal bacterial overgrowth is the use of certain acid inhibitory drugs for reflux, for example. If they are taken over a long period of time, this could induce some overgrowth in the upper gastrointestinal tract. So the other question is the active bacteria in some plain yogurts equal to a probiotic supplement? So actually bacteria in yogurt can be very beneficial in general for gut health. I'm not saying that it's good for celiacs because we haven't tested whether they have those specific activities that I mentioned during the talk, but a yogurt is a nutritious food, and if it contains live cultures, it can sustain overall your microbiome. So it's a good way of consuming live cultures as compared to a pill that you don't know where it comes from. If it's a well-characterized probiotic, you could take it in pills, but sometimes those pills do not contain, if they are not from a reputable source, what they say they do. So you have to be careful.
Mm.
Can a low FODMAP diet mask dysbiosis, SIBO, by not fueling bacteria? Oh, so the FODMAP diet. This is a very fashionable diet in the area of IBS and IBD. I think maybe the success of the FODMAP diet is also overrated. It is a very restrictive diet. It takes out sources of fiber that are very important for our microbiota. And it is not sustainable in the long term. So you need to follow, if you're going to start restricted diets like this, which I don't necessarily recommend, you need to have a very good and close follow-up with a good dietitian or nutritionist that is going to make sure that your diet is adequate. I don't think that the low FODMAP diet is related with SIBO. I have never seen anything on that. So I think there's no relation there, but I could be wrong. Are oral lesions likely related to microbiota deficiencies? Or I don't know what kind of oral lesions you may be referring to, Joe. There are, there are so many kinds of oral lesions, but I don't think it has to do with that unless you have a problem in your oral microbiome of pathogenic bacteria that could, for example, increase gum disease, or, for example, you could have yeast infections, but I'm not aware that oral lesions necessarily are related to a change in microbiome. Does this mean our body doesn't absorb fiber, or we need to eat more fiber? Okay, no, maybe I didn't explain well. So fibers, as I said, are very large carbohydrates. They are plant-based, and it is very difficult for us to break down these substrates. Us, when I say us, it's our own enzymes. We can't do it. We are not like cows, for example. We don't have those enzymes. But the bacteria in our colon does. And so if we eat fiber, we actually can extract the nutritional value because of our bacteria. Okay? So if we don't eat fiber, those bacteria that have the capacity to break down fiber are going to disappear. So yes, it is a good idea to eat fiber, but there are some people who are fiber intolerant. So you cannot just go from 0 fiber to 40 grams of fiber a day, because you're going to be bloated, okay? Because as you digest the fiber, you produce gas. So you have to do this again with very close, maybe dietitian counseling, increasing the dose of fiber and seeing which fibers you tolerate better, because not all fibers are the same. So what probiotics or supplements? I don't recommend anything. I don't recommend anything. I study very carefully, very specific probiotics. And the day that I can find a specific strain that shows in a clinical trial some efficacy, some outcome in celiac disease, then I would tell you. So far, I can just recommend, for example, the question of the yogurt. Yogurt with live bacteria and live cultures is good for your overall health. But there is no specific recommendation for celiac disease. How do we decrease the celiac bacteria and increase healthy bacteria? Okay, so this question has to do with balance of our microbiome, right? So one way of doing this is to eat a diverse diet. So I know you have to eat a gluten-free diet, but eat a gluten-free diet that contains different elements. Don't just eat one brand of gluten-free cookies. Add natural foods that will add that fiber, the tryptophan, and so on and so forth. So this is what you have to do. In other words, don't starve your bacteria. Give them as much diversity in food as you can. Is this study going to bring about a pill for L-tryptophan? So there are supplements of tryptophan. The thing is that they are not necessarily formulated in the way we used it in our study to be delivered in the small intestine. Yes, we are working on a clinical trial, maybe in combination with a probiotic, where we would have this delivery in the small intestine. And if we show any positive impact, that would be the idea. So yes, you are right. I find that a meal high in fiber causes diarrhea. Okay, so this is what I mentioned. Very good question. There are many individuals that are fiber intolerant, and sometimes it has to do with the dose. You are maybe eating too much fiber. All of a sudden, you didn't do that gradual increase to allow your bacteria to adapt to that substrate that you are bringing. But also sometimes it is about the type of fiber that you are ingesting. So you have to do a little bit of a self-assessment of what kind of fibers you can eat. I don't tolerate Brussels sprouts, but I can eat tons of lettuce, which is cellulose as a fiber. So it depends. It depends on your own body, your own microbiome, and you can very slowly educate your microbiome if you wish. But then you have to be consistent. What works, then you stick to that. You mentioned the brain connection. How does it affect the brain? Okay, so these are not my studies, but work in McMaster have shown that certain bacteria produce metabolites that can affect neural function. And the brain, the gut is the most innervated organ, and it has direct neural connections to the brain. So you can imagine that these metabolites produced by bacteria in the gut are stimulating the endings of these sensory neurons, because your gut is sensing all the time what is out there. And these neurons get activated and they send signals to the brain. And so depending on what metabolites are produced, one neuron will be activated or the other. I'm being very simplistic in the explanation, but this is a very important pathway. The other one is the production of metabolites that can actually be absorbed and that can reach the brain and can activate different areas, different pathways. So the microbiome gut-brain connection is bidirectional, and it's an area of intense study. For example, work in McMaster in the Farncombe Institute in the IBS population has shown that certain bacteria can affect depression or anxiety, and that some specific probiotics can improve this communication. How can I improve my tryptophan metabolism? So one way is to consume foods that are rich in tryptophan, and these are usually poultry, broccoli, bananas, chocolate, so good stuff. So again, through a varied diet that is going to provide this substrate to your bacteria and promote this metabolism. Any studies into fecal transplant? So no, fecal transplant is not being explored in celiac disease because it's a small intestinal disease, and you would have to do the fecal stimulation through orally, which is not something that I think many people would accept. Because the fecal transplantation is mainly done from the other end. On the other hand, let me say in general that fecal transplantation is the initial step for modifying the microbiome, and the area is moving into specific consortia. So what in that fecal transplant works? Because when we do a fecal transplant, you are using exactly that. You're using a slurry of the feces, right? A smoothie of the feces of a healthy donor. And you are putting many other things in addition to bacteria. And so that's where the field is moving to. And I don't think that in celiac disease, this is going to be a priority.
I think
I'm
we
trying
have
to
time
see
for maybe one more
one
question.
more question. Oh, I feel guilty choosing one or
I
another.
know.
Um, would baking sourdough with regular flour be fine to eat? But I mean, this question, be very careful. I mean, what kind of flour are you eating? And yes, sourdough can reduce the amount of gluten, but it's not going to make it safe for celiacs to
Mhm.
consume.
Okay, perfect. Well, thank you so much. That presentation was so insightful and we learned so much. I took so many notes, and I hope that the viewers learned a lot as well. We had lots of questions. Sorry for anyone who we didn't get to. If you have any further questions, reach out to us at info@celiac.ca and we can try to answer your questions there. Before we wrap up, again, just want to say thank you to Dr. Verdu for sharing her time and her expertise with us tonight. A big thank you again to our premium sponsor, Odos, for their continued support of this webinar series and for their commitment to the gluten-free community. And before you go, just a quick reminder, we have our registration open for our national conference. So that is November 15th. It's on a Saturday. It's going to be an incredible event with so many researchers presenting there. So it's a full day of research. So you don't want to miss it. You can sign up on our website at celiac.ca. We'll also send a follow-up email with a survey, the registration link, and everything will be in there with some quick tips or some a conclusion basically of tonight's webinar so everyone can access that there. So thank you everyone again for being here. Again, thank you so much. It was a wonderful presentation, so insightful with so much knowledge. So thank you and everyone have a wonderful evening.
Thank you. Bye-bye.
Bye.